So actually, I am able to do this live. We're not really seeing anything right now because I'm working on testing the stream. And what we're going to do, I think, is just breeze through some videos, and I'm going to re-record the videos that are of some interest to me. Just to check a few things out. Let's see if I can do this now. Where are we? Well, this one here looks interesting. Let's see if we can do this here. I'm actually able to monitor my video on another screen now.
Hello, we're on our way for a walk to look for some mycelium.
Alright, so before we do that, let's see here. If I do a window capture, select, okay. Okay, let's see here. Now I'm just going to adjust this here. I know my resolution, everything's all messed up probably, but what are you going to do? Maybe I can put a title on the top or something. So this is just a video. I mean, none of this, I'm just picking this stuff up at random here. And I do have chat monitoring on my other computer. So that seems to be coming in. I don't know how the voice, and I'm just testing sounds and that sort of thing here.
So is this mycelium?
No, this is this flower, but this is how it looks before it comes up.
So I have a situation with my mushrooms, but I'm not going to talk about that.
So basically what we're looking at here is we've got decaying wood, that the mycelium, now this is rhizomorphal, because you can see it's like a root system. Different types of mycelium have different structures, physical structures. And this rhizomorphic mycelium is this rooty kind of...
Now what that means, rhizomorphic, that means, I believe, where the mycelium goes into the roots and it helps. It's like a symbiosis between the mushroom growth and the roots of trees to give the trees nutrients, and it transports nutrients back and forth between the roots and
so what we're going to do is we'll take a
mycelium,
which is we'll put it into a zip
the
bag,
outgrowth of
and then
spores.
we'll try and grow it out in a Petri dish. And we'll see what mushroom it turns into later on.
Wait just a minute now. Hold. Okay, here he goes.
Sample.
Now see, he's just grabbing that with his fingers. And this is, we're not growing I'm not growing mushrooms to eat or anything like that.
Try and get some from...
Yeah, I mean, who knows, you know, I could start a whole project.
No, these are just the physical characteristics of the mycelium.
So what did you call
So this is rhizomorphal, rhizomorphic.
Rhizomorphic.
Those look like dead roots, though.
That looks
Yeah,
like,
this is mycelium.
It looks like elder. So that might be the juicy.
This will all probably be the same thing, or? Yeah, but this is also it, isn't it?
That's definitely
maybe so.
mycelium. Good little video, I guess.
So, when you're finished looking for your
Look at this big log.
try and cover up your mycelium, because of course you want it to not be disturbed by us, and we want to carry it to live its little thing, do its little thing. Cool. So here, Lots of
Now it'd be interesting to note if you could grab mycelium like that and just put it. Well, he says something about putting it in a dish and
Can we take some
growing it right from there, but
something
like
else
he's
to do
handling it with
or-
his fingers and
This is
everything.
mycelium.
But he's put it in a plastic bag which may or not have been sterilized. So I mean, you know,
Yeah, so this is elder, you think?
Yeah.
I don't
So
believe
maybe
that
this may be the.
sterilization
And
is
that's
100%
Mhm.
important if you're not planning on eating any of the
Yeah.
mushrooms.
So where best to take from? Straight from here or from
Oh, no.
here?
Take it from a
Look,
couple
Yeah,
of
parasitic plant. Yeah.
Yeah. It doesn't photosynthesize,
Yeah.
does it?
take it from a couple of places, just so that we've got better chance of.
This much?
It's a parasitic plant.
Yes.
And it doesn't photosynthesize. It takes all its energy from the roots of another species.
Unlike
Kids are
traditional
cute.
mycology, we don't necessarily know what mushrooms these are going to be. But
No
because
kidding.
we're doing research on material aspects of
Oh, that'd be
mycelium,
interesting. Just grab
we
a whole
don't
bunch of
necessarily need to know
mycelium
just yet what they are.
and then
So what we're going to do is take these,
Sprout
these
'em out and see what
samples that we've
grows.
collected. We're going to put these back into Petri dishes, and we're going to grow them out over the next few days or weeks, and we'll see what they become.
okie dokie. Now before we get into that, let me just zip over here. Oops.
What they're doing is
No, that's not what we're doing. I was able to cut that ad. I want to go back here. And let's see, we're going to go to that person's site, look at the videos, and see if we can find, oh, there's other parts there. Part 4, I think was the next one. How to make spore jars, laminar flow cabinets. I'm using a still air type of aquarium setup for now because I'm just getting started into all of this. So, oops, wait a minute, that's how to make agar. Okay, maybe, let's see here. This this is part 4 of that video set. Brewer's yeast. So now, I don't have a pressure cooker yet. They're not really that expensive, but I'm a little bit tight with the money right now, which is not such a big deal. Well, it shows some stuff that he's doing. Let's just see what's going on here. I'm not actually using this procedure, these chemicals. I'm not even using brewer's yeast. Now, okay, so when I set up my solution, which what I'm going to use is called potato dextrose, but and I probably have to send away for some stuff because since I'm starting out with this, I'm going to be trying different control dishes for my environment and make sure that I know what grows in the dish besides mycelium. And if you notice, I believe that's the solution. So she's got it in her hand. It's, everything's like pretty much out in the open. I mean, and there was 2 setups. One setup I noticed where people would boil the distilled water, and then the other would be to do maybe what they're doing is once they have their solution cooked, then take that solution and put it in the pressure cooker, which is in a container, and then sterilize it that way, which I'm not familiar quite exactly how that works. I just know that they set the pressure to like 15, I think, psi for about maybe 20 to 25 minutes, and that sterilizes the solution. Let's see what they do here. Okay, so adding the agar, I guess, I don't know if that's what she did, but if you add that slow, it doesn't lump up. I guess the solution's already warm. Or is it? I don't know. That's a question because the videos that I follow, they heat the solution, well, the potato dextrose solution is just boiled and then it's filtered. And then the agar is added to the hot solution slowly so it doesn't clump up. They may just be using distilled water. So I believe she's stirring in agar. And I don't know what that is. I'm guessing. Okay, so here I guess they're stirring in the mix while it's heating, which is different from another video. There's different ways of doing this, and maybe it all ends up being the same once you get it into a pressure cooker, but the other video that I watched, once the solution was created and put into a flask, that flask was put on an electric burner, and it was, I don't know if it was set to boiling, but maybe just under boiling for, I guess, 15 to 20 minutes. So she mixed that in without it being warm. Now, I mean, another thing, the person doesn't have a mask on. She doesn't have like all of this gear. There's protective covering that you can use. This is not a really sterile. Well, it's a clean environment. It's cleaner than my place. Let's put it that way. She got plants growing. I got some dead plants in my house, but yeah, too funny. So I don't. I'm guessing they just add the brewer's yeast and the agar to distilled water. Transfer the agar mix into the jar. Now, another thing I noticed, it seems like maybe I'm missing something, but I was thinking this should be half full. I don't know. I know that was 100 milliliters. They're using. Oh wait, just a second now. There's a number here. Let's see. They're using 600 milliliters of water and 10 grams of agar. The other video that I'm looking at uses 100 milliliters of water and 2 grams of agar and 2 grams of corn sugar, I believe. So I don't know what ratio that. If I divide this by 5, that's 120. That's close. It's a little bit more water. But I'm not using brewer's yeast. I didn't even know what they were doing here. So let's see. Okay, let me just jump this up here. I'm just looking at different ideas about how people do this. And the importance of sterilization and not having mold and stuff and bacteria grow in your Petri dishes. And this is just a test stream to see how the stream works out. Oh gosh, there we go. His voice turned up. Sorry about that.
Ooh. Looks like aquafaba.
Looks like aquafaba.
I'll show that you can shoot.
Yeah, we want it to have a hole, or else it will explode
Yeah, absolutely.
when it steams off. Okay, so let's go put you in the mix.
Yeah, this pressure cooker looks like. Wow. Looked like it was set
Okay,
at 15.
so here we are outside with the pressure cooker. We're going to put a foil cap over the jar just to prevent any water from dripping back down into the jar while it's in the pressure cooker. The pressure cooker is already
I don't know how important sterilization is
warming.
here. Like, he's outside and
It won't take us
everything.
So long. Dave, just pass me one of those. Matt, just pass me one of those bricks. We just want to.
Okay, so he didn't really show. Another thing is like if he puts just one jar in there, it could be moving around and stuff. You could like put 2 or 3 jars in there to keep it from moving and jumping around and so on. Let's see here. I guess we'll jump to the next video and see how he progresses here. That would be part 5. He's using 95% ethanol alcohol. Oh,
So
wait.
here we are. We're about to.
Wait, so spray inside lid and base of each Petri dish with alcohol. And he's got his hands all over the dish. I've seen videos where, like, I'll be doing this process inside the closed air container with gloves on, and even my container is going to be wiped down. Everything's going to be a lot more sterile, but and I'm only using. You can use 91 or 70% or 90.
So here we are.
I
We're
guess.
about to prepare the petri
I don't know if that
dishes.
really matters.
So what we're doing is we're just giving each petri dish, the lid and the base, a slight spray with alcohol. This is 95% ethanol. This will just make sure that there is no contaminations in there. so that when we apply the agar to the dish, it will last a little bit longer. It won't get any contaminations. So this is a little bit of a cheat. Okay.
I guess so.
Now you'll see that we're stacking the Petri dishes in stacks of 3. This is just easy number to work with when you're moving, filling with agar, closing, moving.
What is the
filling with
purpose
agar,
of this
moving,
guy
closing. You'll see it.
sitting here?
To that stage.
Spray with alcohol and wipe down all surfaces inside laminar flow. I do not have a flow hood. That's why I'm
So,
using.
Before we get started pouring the agar into the Petri dishes, we're going to.
And I'm also using a clear container. You can't see very well through that.
Wipe everything down, arms, gloves, and then before we transport the dishes in, we will clean all the dishes before putting them into the environment, so everything is absolutely clean.
So he's improving his sterilization technique as he goes along here. More or less. My container is an aquarium. It's a glass aquarium. And I'm going to put plastic with gloves that go inside, and maybe somehow I got to be able to transfer that stuff into the aquarium before I do the final sterilization. I sort of have a hood, a makeshift hood. It's not the best setup, but hey, what are you gonna do? Oh, he's got 2 holes here for this, and they're probably just open. I mean, you can wipe all this stuff down, and there's still like. Oh, he's got a filter on this pump here.
We're all cleaned up, so we'll start the pump so that we start pumping.
Oh, that is noisy. And that's another reason why I'm not doing this airflow method, is because I don't really want to listen to all that.
The idea of this pump is to flow air, so we can see that there's air coming out through here and through here.
I was running the filtered air
So
through
this is showing us
the.
that there's clean air being pulled in, and we can work inside this environment without any contaminants.
Okie dokie. Wipe external. Dish. Okay. So what he's doing now, he's wiping his dishes out here and then putting them inside, I guess. I don't see any problem with that. That kind of eliminates the purpose of having the holes here to begin with, but he's doing his transfer inside, of course, later.
That we just made, and we're going to clean this also with the alcohol, just to make sure that everything that goes in there is clean.
Okay, goes the alcohol swab down on the floor. Now this is another thing that I don't like. I don't like the idea of just spraying all kinds of alcohol all over the place. It's just smelly, and God, what if somebody lights a match? This is plastic. Everything is gonna go up in flames, and that's one thing that kind of bothers me is the smell and everything. And I'm thinking, I've got my setup next to a window. I was thinking about putting some kind of ventilation and running it outside the window. I don't know exactly how I would set that up with a glass container, but I wanted to do like sort of a double, I wanted to have like a clean room, sort of a small clean area, and then have the terrarium connected to with a little hose. But I don't think that's gonna really work. I mean, for what I'm doing, for the purposes of just starting this mycelium growth, my daughter says your place is not conducive to grow mycelium, and I want to prove her wrong. And it's gonna take me some time to do that.
Just be careful with this because the jar can become very slippy.
Slippery.
And have a tendency to...
God, I would not want to drop that jar.
Make sure you get into these crevices because that's where you're going to be opening it from. Okay.
I have not seen this video.
Okay.
I kind of like it. I mean, it's pretty close to an idea that as far as transferring fluid into the Petri dishes, what I would do. Now, so supposedly all the air is going out of those holes, because that's the only place it can go. So there's no airflow into the holes, and because he's wiped down the sides, it should be a fairly sterile environment. That's interesting. What would be even more interesting is like if I had a pump set up to run air through a tube where I don't have to listen to this pump and just run the airflow through the tube and run the tube into a filter setup, and that filter setup has to be like down to 0.2 microns or something. I could do something like that. I could have an enclosure outside. Maybe, I don't know. Because you might, whatever. I think it's a good idea. I have to listen to
So the
that
idea
noise.
behind this is to work as quick as possible. So we
Right.
want to be able to basically lift, pour, close, move, lift, pour, close, move, and just keep the process as quick as possible so you don't get any contaminations into the Petri dishes. Okay.
I wonder if you could do that with the cotton on top of the flask. There's a 100, well, there's a 250 Erlenmeyer flask. I was thinking maybe getting that so I can have that. That would be my final transfer of solution rather than a jar. And the jar, the flask, the other video, I could show you that video. I think I know where that video is. We could run that video. That'd be a good comparison. And there's like sleeves you can get to cover your arms, and there's the doctor's face mask and the head gear and the whole body suit and everything. He's not using any of that. You know, he just got the gloves on. I have the same setup pretty much. Now, well, another thing too is I don't like handling that big jar inside. I think I would be much more comfortable holding an Erlenmeyer flask because it's narrow at the top, and there's something else too in that other video. We'll watch that other video. Oh, his jar is open. He's leaving the jar open. And the way he's doing that, they could also end up getting bubbles in Petri dishes. Well, let's see what's going on. Place field to one side to cool.
Okay.
Oh, wait.
So once we got those Petri dishes, we can leave them on the side now just to cool down. We don't want to open up the Petri dishes anymore.
No.
They need to be sealed until we're ready to inoculate them.
And there's something else.
You want basically around about a millimetre of agar on the bottom of the Petri dish.
I I was told have enough fluid so that the fluid level comes up to the level of where the top of the edge of the cover is.
Because that's all you need for the mycelium to colonize the...
Wait till you see what solution I got conjured up. Now, I don't understand, you know, for me, I would let, he said to let them cool. Well, I would let, and then he's got a plastic dish here. I was thinking about maybe having them, I'm going to leave these dishes inside the container here, and also use, there's a type of tape that you can put around the seal to seal the edge of the dish, because if you don't, then contaminants can get in that way. I mean, these spores are like freaking micrometers small. Okay, so that's the end of that video. Inoculating up next. Is that... that's not the same. Okay, wait just a minute here. Oh, we don't want this stuff showing up here. Let's see here. Let's go to the next video in his series. 5, I'm assuming how to inoculate dishes. This was done a year ago. And I'm not just working with mushroom spores, which is going to be interesting.
So now we're ready to inoculate the Petri dishes with some of the mycelium we collected this morning, and we have some mushrooms that we bought from the supermarket.
Oh,
so we're going to take
the
tissues
supermarket?
and transfer them into the agar. Now I'm going to show you this not in the clean box because this is already a contaminated Petri dish.
I didn't think
But
you could
basically
do
what
that.
you want to do is...
I wouldn't want to eat them.
You're going to take a tissue sample. So the first thing you're going to do is sterilize the blade. Okay. Then to cool the blade, just dip it into the agar. Okay. Then take your tissue sample and then transfer that.
I
into
saw
the
that,
agar,
but
you
now
just
he
need
just opened up that dish and all kinds of contaminants just got in there.
So you're not
Well,
getting any cross-contamination.
yes or no?
Okay.
No.
So
Never.
sterilize.
You have to do that every time. Now,
Then
see, this is another thing
cool.
that really bothers me, is that he's got all this alcohol smell, and he's got this flame burner just sitting right there. That's why I was opting for a completely enclosed container, so that you do not smell the alcohol anywhere. Hopefully, making the flame situation, area, whatever, a lot safer. Maybe I could even have some kind of a slightly enclosed container for that too. That makes me nervous.
Then tissue sample, Petri dish, and then repeat the process. Okay.
And that
That's
look
all
at the
we
wind
need.
blowing across that flame. Dip, place tissue, Petri dish, close lid, rinse and repeat.
So we've got various agar mycelium that we found in the forest this morning, and we're also going to take tissue samples of various mushrooms. We've got shiitake.
Oh, this is 12 minutes long.
Oyster and chestnut.
And white button.
And white button mushroom. Okay, so we'll take these tissue samples and put them into Petri dishes, and we can go on from there. Okay. Right, so Dave, do you want to switch that?
I mean, he's obviously had success. He knows what he's doing.
As always, sterilize everything.
The flame burner is inside? Oh my god! Are you kidding me? I just would not dare to do that. I would just be so afraid that that whole container would just light right up. There's just no way I would do that. I only have one house. Shit.
Well, if you're taking a mushroom like this, you can use the hot blade to make the first cut on the mushroom. If you're gonna... I'm talking too loud. Oh, I'm talking quietly. Okay, sorry, I've got my earplugs in. I can't hear a thing. Okay, so we're going to use a hot blade to cut the mushroom. Okay? Some people say it's best to break the mushroom up and cut it, because that way you're not introducing your blade into the thing. But I found that if you use a hot blade to initially cut it, so keeping the Petri dishes in the clean air.
I did not see him wipe his gloves down with alcohol. I mean, wow.
We're now just going to fill the...
Look at that flame is bouncing around everywhere.
We're going to be very careful here to keep the scalpel very still as well. And you're just going to take a tissue pencil and you're going to put that into your tissue.
Now there's another step here that he didn't do, which involves taking the section of mushroom and dropping about 4 or 5 sections into a small dish of hydrogen peroxide, which sterilizes the contaminants around the mushroom piece before putting them on top of the Petri dish. So I watched a video about doing that. I think that's a good idea. And maybe if I do that, I can totally avoid having to have the alcohol burner inside of this container. Because I'm using like I'd have the burner outside, treat the knife, and the knife is now inside the enclosed container, and it's only working with the same mushroom. It's not going from mushroom to mushroom. And then you're going to take those samples and put them in the hydrogen peroxide. I mean, it seems to me that's a lot safer idea.
From the same mushroom in a slightly different location, here's another sample.
God, what if that flame just grabbed a hold of the alcohol in his glove? You know, like that whole glove would just light right up. That just scares the living crap right out of me. Let's check this in here.
So somebody wants to write on what that mushroom was, put on, and put the date on.
Okay, what is that bag? Wait, let me... What has he got here?
Samples that we found earlier.
Oh, he's got the samples that he found. Okay.
Here's the sample, you can see it.
Now, collecting a sample like that off the wood, that's going to have, like, supposedly all kinds of stuff on it besides that mycelium, but he's obviously done this before and has had success with, I guess.
Firing in there.
It's not what I would do. I mean, preferably.
So somebody wants to write on what that mushroom was, put on, and put the date on.
Alcohol just spraying everywhere. Oh, heavens to bitsies. He didn't sterilize the tweezers. Well, Theoretically, the mycelium he's got's not sterile anyway, but you don't want to add more stuff to it. Oh, he's just putting it right in the dish.
You want to slightly tap the mycelium down into the agar when you place it in the petri dish, just so it doesn't have to go searching for food. You want to get it to establish itself as quick as possible.
Well, I suppose tapping it down into the dish a little bit, I don't see a problem with that. You just don't want to slam it down into the dish, maybe. I've just seen people just drop the stuff in there and don't touch them at all.
So, you just put a base in that. Trust your mind, I don't know what it is yet.
I'm new to all of
so
this.
we're going to demonstrate this outside the box because the box makes an awful lot of noise, it would seem. So we're going to go through the process that you would see us doing inside the box. Whenever you're transferring mycelium material, there's always going to be a chance that you're going to get contamination. So you may have to repeat this process 2 or 3 times before you get a clean mycelium. So first of all, you're going to sterilize your
Right, so what he means by that is, so you get the mycelium in the first dish, it's got a bunch of stuff in there, plus the mycelium that you want, and then you take that mycelium out, transfer it into another dish. It's sort of like a way of filtering out the contaminants. So I know all about that.
blade. Okay. Then place it into the agar that you're going to be using to transfer your sample into. Because what you don't want to get is any cross-contamination. So you're always doing the same thing with the same petri dish. Sterilize, work with the petri dish. So we're going to take a small tissue sample. It doesn't have to be large. You can just take a small tissue sample from this. Okay. And we just place that into the petri dish. Okay. You want to tap it slightly
Oh,
down.
he's putting it on the edge.
So it's in contact.
Why not
with
put
the
it in the
petri dish
middle?
So it has access to food. In a petri dish this sort of size, I reckon you can do about 3 to 4.
I see.
Probably ideally 3. Of
I
the
see what
same
he's
mushroom, the same tissue type, in 3 locations.
Oh,
Because you may
okay.
find one works slightly better than another. And always try to collect the tissue sample from a different location on the mushroom. Because you may find that in a certain area it's more alive.
Okay, that's a good idea too.
yeah,
So there's some good ideas going on. See, I was just thinking,
So just tap that
I was
onto
just
the
thinking,
tissue.
okay, I used one dish to do the whole thing, but I don't see a problem with doing that.
And that's it. Sterilize and
He's
do
got
the
3
next
in
one.
there, 3 different pieces. Now it's going to conclusion of the workshop.
Okay, so now we have our petri dishes. They're all inoculated with various types of mushrooms. Some of the wild ones that we picked this morning, some of the ones we bought from the stores, and we've got a pile of mushrooms here, which I now believe we're going to take to the kitchen and cook for dinner.
Yay! So how long will it take for these to grow out?
Okay, we should start seeing some life in 48 hours. Okay. Then we've got to keep an eye on them for contaminations. If we've got enough of them, then it's up to you. Either we extract the mycelium away from the contamination, try it again. It's good practice in any case to do it. Or we just remove that one. But it's we've got plenty of working ahead of us. We've got to now monitor these. Get a nice strong sample before we transport that into the spore jars that we created yesterday.
Okay. So after 48 hours you would see some growth, and then how long before you could transfer it?
A week and a half, something like this.
Right, okay.
Yeah. Depending on the growth speed of the various mycelium, different mushrooms will have different growth rates. It'll take 48 hours before you start seeing it. It will start coming out like very furry tentacles outside of the mycelium. If you start seeing other blotches of growth, that's probably contamination. They'll be in a completely different area to the mycelium. The mycelium, you'll notice
Is
in
white.
certain scenarios that
I guess all
if
mycelium
there
is
is
white.
contamination near it, it will be killed off. So you'll see the contamination being killed in the pattern of the mycelium because it will start producing its own antibodies. So yeah, it's an interesting thing.
And we're looking for the fast-growing ones.
Well, we don't ideally, as a material for construction, it's going to, we're going to have that balance between space that is taken up while the blocks produce and speed and strength of the material. So we may have very fast growing mycelium, but not very strong blocks. So they could be used for insulation or these types of things that are passive. Yeah. Where we may want very strong mycelium, very strong blocks, which may take a little bit longer. So we've just got to try and find a balance between.
That's interesting what he's saying. I wasn't sure. I didn't know you could use that to create insulation. Faster growing mycelium is a better insulated, has better insulative properties. I do know that the blocks, some of the videos that I watched, they were fairly fire retardant, which I thought was pretty impressive. And they're making construction materials out of this stuff, and tables and chairs. I never knew any of this. It's pretty cool stuff.
Because we've also got to store these blocks while they're forming. Yeah. So if we've got to store them for 3 months, that's a lot of storage space if we're going to have 3,500 blocks for a house. Yeah. And if we want to store 10 houses, that's a lot of storage space that we've got to find for 3 months.
I tend to kind of go off the beaten path here, which it looks like I might do right now because I'm watching this. Just cut this ad here. We can just get rid of that for a moment. All right, so sorry about that. Let's see what we got here. I want to check this out here.
Hi, my name is Maria Peñil. I'm a multimedia artist and I've been working
This is
with
what
Mena
actually
for the
got
last
me
6
started.
years making bacterial art.
Believe it or not, I watched a video about rain hitting a mushroom and millions of spores are coming out, and it was creating this interesting art background of the millions of spores. This is what got me kind of started in all of this, and then partially, and then the other part of why I'm doing this is because I'm interested in looking at spores that are millions of years old and possibly trying to extract enough DNA to bring these ancient spores back to life. I do have a bunch of plant fossil mud. That's why I'm not just looking at mushrooms. I'm looking at spores from different kinds of plants, such as moss and ferns and that sort of thing, and studying growth rates. There's a fair amount of. It's more for the science of studying the different types of spores, how they grow in different types of mediums. Nothing too particular, but I want to see how the relationship is between spores that come from ferns and mushrooms and how that mycelium differs. I have no idea.
15 ASM's Agar Art Competition, both the first prize and the popular
Oh, look at
People's
that.
Choice Prize. And today we'd like to show you how to make Agar Art using bacteria. Come on.
Well, they're using bacteria, so they're not using the music's loud. Not spores. They're using. Oh, I know what they're doing. So they're just spreading the art across the dish.
Here we are in my lab.
Or the bacteria.
Before we start, we should suit up. You have to wear a lab coat, protective eye gear, and gloves. Make sure to have no open-toed shoes. For a complete set of rules, please visit ASM's website. In order to prepare our 1 liter of agar, we need to weigh 15 grams of agar and add it to the bottle. We then need to weigh and add.
So 15 grams, that relates to 1.5 grams and 100 milliliters, according to this particular video. And I don't know what peptone is. So they're using an agar peptone mix. Now I'm going to stop this video for a moment, and what I'm going to do is, what am I going to do? I'm going to go look for Bear with me here. Why is that doing what it's doing over there? I don't know. It should be... I don't know what my stream is doing. Let's see, I'm gonna look for a web page. I'm gonna try to get the back of the web page on the video here. Let's see. Clone mushrooms, tissue culture. Here it is here. Favorable cultural conditions for mycelial growth of Korean. No, I don't know. I should be able. Let's see if I do this. Now we can see the web page, but for some reason my stream over here is doing something awkward. So I'm going to just renew my stream and see if I'm still streaming here. Okay, good. I don't know why. Alright, so now what we've got here is this is an abstract from a mycology study that was done in Korea, I guess. So we're going to go down here and we're going to find there's the different types of media. PDA is your potato dextrose, which is what I'm going to do. They're using peptone. Now here's one called tryptone. Agar is part, that's your base medium. And they're using, okay, here's the peptone down here. So there's the GP, the MC, and the YM. Those are the names of the types of media growth. YM is yeast malt extract. Glucose peptone. That uses malt extract and glucose and yeast extract. I don't believe that's what they're doing. I think all they're adding is peptone. And in both cases there's a malt extract added. So these are and I'm using potato dextrose because it's easy, and what I'm also going to do is do two different types of, like the first growth medium will be potato dextrose. I'm looking for the corn sugar. I haven't found that yet. The second one is going to be potato dextrose, but not with the corn sugar, or I'm going to do a half and half of corn sugar and honey. I think that's better. That way the new strain is creating different enzymes in order to survive off the new types of sugars. And I was told they grow better if they have different types of nutrients that they can get their energy from. And the other is sucrose, which is just plain table sugar, and they're actually using that here. So now if I go, the sucrose is added to the agar, but it's not added to the potato dextrose. Now potato dextrose, I'm assuming potatoes got the dextrose already in it. The corn sugar has dextrose in it too. So they're really compounding the dextrose in that solution. Oh wait, the potatoes are down here. With the sugar has fructose. Oh gosh, I can't even remember now. Let me see here. can't see some of this stuff. So table sugar has, well, they say also known as dextrose, but glucose and fructose is table sugar a mixture. Let's see. Sugar is a compound. Let's see here. Sucrose is a composition of carbon, hydrogen, blah, 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 blah. Two monosaccharides, glucose and fructose. So, but the glucose, the glucose, what did it say? Includes glucose, also known as dextrose. I don't really understand the types of sugars. But you know, I'm just going to use different, I'm going to try some different mediums and check out my environment, all that good stuff. So we go back to here and we go down through this list. These are the different, there's effective pH on mycelial growth. I am not taking that into consideration yet. I'm guessing the pH is going to be around 7. It's not, it's supposedly in the middle, and there are methods to use to balance the pH if that's something that needs to be added, which in my situation as a hobby, I wouldn't think so. The temperature, 20 to 25 degrees, they're saying 25 degrees C. So if you look at the numbers, basically, I mean, you've got a double amount of growth here from 20 to 20. So basically, a more warmer environment is going to get you better growth, even up to 30. But in some cases, it goes down in some particular strains of mushrooms. It doesn't grow quite as well. So 25 degrees. Then we have the types of mediums. Now, I don't see, if we go back up to the top of the list and see sucrose here, sucrose is only in the CZA, and they are adding some other components of to add to the growth here. Some phosphates, calcium chlorides, iron sulfate, potassium chloride, magnesium sulfate, some sodium. There's possibly pH balance stuff in there, and some other nutrients. So that's CZA with sucrose, and with PDA there's only the dextrose. So if we run back down here, you can see the levels of the growth of all of the different types of strains with sucrose and dextrose, fructose, and glucose. So if we look at the general numbers, fructose seems to be low on the list for growth. Well, according to that strain, it's not, but glucose is pretty good all the way around. It looks like the levels are lower for the most part. Dextrose, the levels are higher. In most cases than either the fructose or the glucose. If we compare this to the sucrose, obviously the dextrose is the better, I think. So if we look at the numbers on the bottom, the mean average, dextrose is pretty high on the list. In fact, it's actually the highest mean average. So that's why in potato dextrose is really, you know, I think that's going to make a really good base, but I don't have the corn sugar. I can't find it. All I can find is the sucrose. So if I'm thinking, if I substitute the sucrose for the corn sugar, since I already have the potato dextrose, you know, and there is glucose or dextrose in this sucrose table sugar, my amounts are going to be comparable. They're going to be like less. Maybe I'm going to be in the 35 range for average growth for everything here. That's my guess. So I just wanted to add that list in there and show you that I am studying all of these different types of mediums. Now these are the different nitrogen sources. Let's see here. Arginine is right up there. That's ARG. And I've noticed that some of these sources are in the baby formula, but with baby formula also has lactose, which is a problem because mycelium does not grow in lactose. So, but anyway, that takes care of that. So let's go back to here, I believe. Now they're using peptone, aren't they? Agar and peptone, 10 grams in 1,000 liters. That's a medium. Now let me just see. I think there's a peptone here. They're using 10 grams. It's half of the amount of agar. And in the MC, that's GP and MC methods. So if we go down to there's the sucrose CCA. Those numbers are pretty high. Those are really high. Potato dextrose. I don't see why I couldn't just use table sugar. I really don't. I really don't see why I can't. I may just do it anyway. Just say the heck with it. And do like, instead of, so if you go back up to this list, they're using 200 grams of potatoes and 20, but we're going to go look at that video. That's like 10%. Well, no, the agar and the dextrose is the same. And then later on, I'm going to take the dextrose, which is the sugar, and I'm going to split that value in half and add honey to that mixture. And giving it the extra sugars and that sort of thing, I guess. So where's the peptone? I wanted to see, well, the growth rates are for GP, MC, and YM. GP is 76. I mean, look at all these growth rates. They're in the 70s anyway,
10 grams of peptone, 5 grams of yeast extract, and 5 grams of sodium chloride.
Oh, they got the
We fill
yeast.
the
They're adding
bottle
the yeast. So that's
up
your
to
malt
1 liter
extract.
of water, and make sure the bottle is not full all the way to the top. And make sure the cap is loose to allow hot air to escape.
Okay.
Put the bottle into an autoclave and sterilize it
I don't have an
for
autoclave.
30 minutes at 121 degrees.
21 degrees Celsius?
It will come out hot and molten. Be sure to wear gloves and put the molten agar into a tray so that you can move it to the lab.
Wow.
And put it in a safe place where no one will disturb it. Wait for about 30 minutes to an hour for it to cool down.
I like the way she's handling
ready,
this stuff.
When it's uncomfortable to touch, but not scalding hot. Once the agar is ready to pour, hold it with one hand, and with the other hand, lift the lid. Pour the agar.
Okay, now they're doing something here. They're pouring it like really fast, one thing. And number two, the lid is has not been sterilized. But, that's minor because they're in a more sterile environment anyway.
Slowly, make sure you don't form bubbles.
Yeah.
Close the lid and wait for an hour for the agar to solidify.
An hour.
Then turn them upside down and wait for a day for them to dry, making sure that there's no condensation on the lid.
Oh, that's something I didn't. Oh gosh, let's get this guy out of here. That's something that I didn't understand from the previous video.
This is what our finished plate looks like. We're now ready to make some agar art using bacteria.
No, where are we?
Yeah.
Oh my gosh. I don't even know where I was now. I was right over here. So I didn't know about turning the plate over to make sure there's no moisture.
Will disturb it. Wait for about 30 minutes to hold it with one hand, and with the other hand, lift the lid. Pour the agar slowly, make sure you don't form bubbles. Close the lid and wait for an hour for the agar to solidify.
That's a big dish.
Then turn them upside down and wait for a day for them to dry, making sure that there's no condensation on the lid.
Now that's
This
another
is what
step.
our finished plate looks like. We're now ready to make some agar art using bacteria. You can use microbes that are genetically engineered to be colorful, such as E. coli that produce chromogenic proteins. These are available from a number of companies, or you can even make your own using bacterial transformation. There are other bacteria that are naturally colorful. In this case, we're using Xanthomonas from NEB's collection, or these can be purchased from a biological supply company as well. We also use environmental isolates, but that is not recommended unless you can be absolutely sure of what you are working with. For example, we sequence our isolates to find out what they are and make sure they're safe to work with. Remember, of the 30 million bacteria that are out there, we only know of 70 species that are pathogenic to humans, but you don't want to accidentally grow something that can make you sick.
Those are my brushes. They are platinum loops, and usually I have 3 of them. This one is it has a bigger loop, so I use it to get more bacteria and do bigger surface. This one over here is really thin. I use it to do details, and this one is in the middle. Let's get started. Today I'm gonna draw a dandelion.
Now she's using a
and
burner,
I'm
which is a
start
much hotter
with
flame.
Sterilizing my loop.
Wow.
Cool the loop. Take a little bit of bacteria. And I'm gonna draw this stem. Sterilize again. For the leaves, I'm gonna use.
Oh, you see what she did? She cooled it off on the side.
I'm using the medium loop. And for the flowers, I'm gonna use a different technique with a pipette.
Huh.
So I have the bacteria here in rich media. Take a little bit of that.
Now that's that tool I need right there, because I've got all those pipettes. Oh, look what she's doing.
And that will be it. So now I have to let it dry for a little bit.
Parafilm. Okay, now that's what I was looking for. I gotta look for
Now that I'm done with the
parafilm.
drawing, I put it in the incubator for 24 hours. So here is an important tip. Bacteria grows at different rates, different temperatures. So the more you know about the bacteria that you are using, the more control you have about your art. So this is after 24 hours in the incubator, and now I'm gonna let it sit for another 48 hours at room temperature. Okay, my piece is ready finally, and now
That's
we're
pretty
gonna
cool.
show you how to take pictures so you can submit them to the ASM Agar contest.
Oh, there's a contest? Pretty cool.
Here at NEB, we'd like to use a light box. In this case, we can change the intensity of the light, making it brighter. And you play around with the light conditions, neon lights, sometimes outside lights, until you're happy with it. And then we try to stand on top of the agar straight up, like this, focus, and take different pictures. Once we're happy with it, we submit it and keep our fingers crossed.
Okay, so that was it. Thank
So I am just going to make a note here, because that is something that I need. I want to write this down here. I got tips here. Okay, so parafilm around Petri dishes. And what was the other? Oh yeah. Erlenmeyer flask. Boil method on hot plate. I'm thinking I want to go that route because I don't have a pressure cooker. That's sort of my problem there. Now I'm going to look for, we're just going to take this off, and I'm going to look for the video that I used to do that I'm going to do my actual spore production with.
Expect. If you look here, you can kind of see that the top of the media falls roughly along where the edge of that plate is, and that's a good sign you have the right amount. It's 25 to 30 mils, but rather than measuring that exactly, you can just pour until the top of the gel is roughly even with the bottom of the lid. So once you've done pouring your plates, you just want to let them sit on the counter for about 20 minutes until the gel solidifies, at which point you want to immediately flip them over, because as you can see there's a fair amount of condensation on the lid, and if we were to just leave this would drop on the plate and ruin it. So after about 20 minutes the gel will be hard enough that you can flip it. Now obviously it's quite important you don't do this too early, and again I'll show you this with my sacrificial plate, but yeah, if we flip them too early, as you can see, it just spills out everywhere and makes a mess. So that's how you would make wort-based agar, but what about this potato that I've been lugging around? Well, I'm going to use this to make a classic media from microbiology called potato dextrose agar. This is often used for growing bacteria such as Lactobacillus and Pediococcus. It's also used for growing fungi, which would include yeast. And the nice thing about it is it's dirt cheap. I mean, you literally need potato and corn sugar to make this. So what you want is about 50 grams of the potato, which isn't very much. Look at that, dead on 50 grams. I'm getting good at this. We're then going to shred this, add a cup of water, bring it to a boil, and let that simmer for 20 to 30 minutes. At the end of that period, you want to filter this through.
I'm using distilled water, and I'll have
a fine mesh sieve. If you want to go a little bit farther, you can run this through a coffee filter after. The first filter should be through something a little more coarse so it doesn't gum up. This should give you about a cup of filtered potato extract. You need about 100 mils of this, and to this you want to first add and mix in the dextrose, the corn sugar. This will dissolve quite quickly into the hot liquid, and it's then ready for the addition of the agar. Now the agar you need to add quite slowly. It'll clump really readily in this hot liquid, and if it clumps it's going to be very hard to get smooth plates. So swirl constantly, add it slowly, and you should be able to blend it in without significant lumps. Once everything is mixed.
See, he's boiling this on an electric hot plate. It's not being pressure cooked. It doesn't show.
Plates, so swirl constantly, add it slowly, and you should be able to blend it in without significant lumps.
He's not showing the top of the
Once everything is mixed, bring this to a boil for 5 minutes, or sterilize it in your pressure cooker, and then pour the plates just as I showed you with the dry malt agar plates. So
now in that situation, I want to show you. I want to go back here.
And you should be able to blend it in without significant lumps. Once everything is mixed, bring this to a boil for 5 minutes, or sterilize it in your pressure cooker, and then pour the plates just as I showed you with the dry malt agar plates.
Now I don't know. See, what he does is he takes his flask and runs the lip of the flask over the flame really quickly to sterilize the lip before pouring the fluid, and he's doing it slowly to prevent air bubbles. So that's the other step that was missing in that other video, both videos, where the lip was sterilized. So I'll probably do that. I'm wondering about how long he waits. Now with me, I don't have agar, so what I'm going to be attempting to do is I'm going to attempt to use gelatin, and I'm going to use a lesser amount of water with the gelatin to get it to become a little bit more of a stiffer type of agar type mix, so that it's not so flubbery. Um, that's that's what I'm going to be trying, and it'll be interesting.
So it's been a little under a week since I prepared these plates, and I just want to show you the difference in growth between beerwort agar and the potato dextrose agar. So here on the left we have beerwort agar. We have at the top Saccharomyces. So this is just WLP001. Here we have Clocacara. This is an oxidative yeast that would be present at the beginning of a lot of wild ferments. At the bottom we have Brettanomyces. This is just the Y yeast, Brettanomyces bruxellensis. And on the left we have Lactobacillus, and again this is the Y yeast Lactobacillus. So I'm just going to flip this plate over here so you can get a better view of the growth. And I think what you might appreciate is that while there is growth on each quadrant, the growth isn't that intense, it isn't that thick or florid. So these plates do work really well for growing these organisms, but the growth is somewhat slow, and it's not quite as aggressive as we might see in other plates. And I think you can appreciate that just by looking on the top side at the potato dextrose agar plate here, which if you recall I cast on the glass plates. And so here we can see Saccharomyces, Clocacara, Brettanomyces, Lactobacillus, same source, same size loop, so the same volume of cells were plated down. And you can just tell from the back side there's a lot more yeast growing, or bacteria as well, growing on each of these. And so here you can see I did a pretty bad job streaking out the Saccharomyces, but there's still quite strong growth, and there's quite strong growth on all the other quadrants. So aside from being cheaper, the potato dextrose agar actually gives us better growth under a lot of conditions. So just lastly, I want to show to you that these plates are in fact clean, even though I only boiled these medias for 5 minutes. And so here is the other potato dextrose plate I made. You can see from the bottom there's clearly nothing growing, and even if I flip it over and pull the lid so you can get a good view, there's no growth on that plate. And this is 6 days at room temperature. Wow. Since casting the plate. And again, showing you this again, here is the wort agar plate, and you can see both from the bottom as well as from the top that there is no noticeable growth, again despite the fact that these are over a week old.
So anyway, that's the story with that. I will just go, where am I going to go with this? Oh, let's see, what do we got here? Mushroom production, tissue culture production. Oh, where did I see? I think it was this fella's video here. Let me just, I'm just going to do this right live here. We'll go to videos. I don't know. It was, how many, this is yeast lab, this is a yeast lab. Let me just go ahead and we're gonna drop that out for a moment. Oh, for heaven's sake. Let me look at my list. Okay, how to clone mushrooms. It might be this one.
Hey guys, today I'm going to show you how to clone a mushroom on agar. This involves taking a small sterile sample of mushroom tissue, of a type of mushroom that you'd like, and placing it onto agar. This allows it to grow out and allows you to isolate a clean strain of a desirable type of mushroom. The mushroom I'll be using is blue-grey oyster mushrooms because I find they grow very well in our climate. Right, to start off with, you need a food source that also provides nutrients. I'm using light malt extract, and you also need the gelling agent, which is agar, and some food colouring helps to see contamination. Right, my recipe is 2 grams of malt extract per 100 milliliters of distilled water, followed by 2 grams of agar, followed by about 10 to 20 drops of red food colouring, blue also works. This is followed by heating and strong stirring to get it all to dissolve. This is then dispensed into jars, not agar plates, because pouring agar plates requires you to have a very sterile place to do it in, like a laminar flow hood, and I only have a still air box. These are the jar lids are then covered with foil with a hole to minimise the contamination window, then covered with another layer of foil, and then 2 layers of micropore tape. This allows the culture to breathe. Now if you leave the jars like this, they will immediately contaminate, so they now need to be sterilised. I do this by loading them into my pressure cooker. The pressure cooker is a 12 psi pressure cooker for 25 minutes to half an hour. I usually go for half an hour, better to be safe than sorry. But yeah, once you've got it cooking, you need to go out and find a tissue sample. Young mushrooms or pins are best, as they have the most virulent growth. Cut off one you like, and dice it up. Make sure you only get the inner tissue and not the gills, because you don't want to get spores. You want to get a clone of the existing mushroom, because you know that strain fruits very well. Now, because the tissue samples were taken under non-sterile conditions, if you're putting them into an agar, it will most definitely contaminate, so you need to sterilize it in some way. I do this by dropping them in 3 to 9% hydrogen peroxide, doesn't really matter, and leaving them there for about 20 minutes.
So 20 minutes in the hydrogen peroxide, you said 3 to 9% or something? I don't know. I don't know what mine is. I should probably go look. Take a look and see what my percentage of H2O2 is, I guess. Okay, so I do have the 3% hydrogen peroxide. 20 minutes.
This makes sure they are sterile and any mold spores or contaminants on their surface is killed, but the mycelium in the middle survives, or the mushroom tissue in the middle survives. But yeah, so I just open up the agar plate and use some sterile tweezers to drop in the small cube of mushroom. I then repeat this 3 or 4 times. It really doesn't take long, and I'm doing this in a still air box, which I sprayed out with a liberal amount of surface cleaner, which I find works very well. Now, I completely forgot to get any pictures of these finished agar plates. I had quite a bad cold, so this is once they've colonized, I'm then transferring them another generation onto some lovely blue agar that I made. This is me just cutting a small chunk out of a colonized plate and dropping it back onto some blue agar. This is made by the exact same recipe. I'm also making some culture slants. This will hopefully allow me to store this culture in the fridge for quite a while. As you can see, I just cut a small chunk of colonized agar and place it into the slant. They will then be incubated for about a week to 10 days at 20 to 24 degrees. I find 20 degrees is good. It isn't the fastest, but it seems to work. As you can see there, I experimented with very small pieces of tissue, but as I had lots of the plates laying around, as you know, I made 4 of them. Reasonable sized squares, I don't know, half a centimetre to a
He just wiped his tool down with alcohol. He didn't use a burner.
Apparently work quite well. Yeah, as you can see, I made 6 more plates, and you can do them really quite quickly. The most time-consuming thing is actually pressure cooking the plates, because I like to watch the pressure cooker, because it is a jiggle top, and you have to occasionally adjust the power setting of the cooker. But yeah, as you can see, they are all clean. The glove box worked. These are only 3 of them, but the other 3 didn't contaminate. As you can see in the one on the left, the mycelium was let to run a bit too much and it grew up the sides and also ate the colour out of the agar. The agar's gone sort of reddish brown, beige, as the colour, food colouring, was eaten out of it. But yeah, what you choose to do with them now, well, you can do whatever really. You could cut them into cubes and use them to seed spawn jars. That's good, but it does require you to open up every individual spawn jar, cut a chunk of agar and do it. So I find the best thing to do is to make liquid cultures. This means you only need to cut one small chunk of agar, put it into a nutrient liquid, and this allows you to easily make literally as much spawn as you could ever want. But yeah, as you can see, I also made 2 culture slants. I let these grow for a week and then sealed them in a Ziploc bag and refrigerated them. The Ziploc bag isn't completely sealed, so don't worry. As you can see, the culture slants do have a hole in the top covered with layers of micropore tape to allow them to breathe. But yeah, these 6 jars, they worked. I used malt extract agar, as I said, but if I'm going to do another generation, I'll mix it up and I'll add 1.7% honey. So it's 1.7% honey and 1.7% malt extract. This is because honey has a wide variety of nutrients and sugars. This means that the mycelium doesn't develop enzyme selectivity, meaning that it only eats malt extract.
Now see, that's one of the other points. He used a half and half mixture of the honey and the malt extract, so that the mycelium doesn't, it makes extra enzymes, has an easier time to grow, I guess.
And will be really slow when it moves on to another substrate. If you use a little bit of honey in the mix, this means it retains the enzymes to digest basically every type of sugar. But yeah, hope you enjoyed, and I'll see you in the next video.
So I guess that's pretty much it. Now what I'm going to do is, because I'm studying spores from ferns, let's see, grow ferns from spores. Okay, yeah, this is, it was this video that I was watching here, so let's do that one.
Hi, my name is David Parasso. I've been propagating ferns from spore for over 5 years, and some of my friends suggested I put together a video on how to do it. In addition to this video, I've also put some information on the website of my micronursery, 4pnatives.com, and I encourage you to read it, as it has many details that wouldn't fit into a video of this. Sporangia spring apart and launch the spores into the air. Before the spores are ripe, the source is often covered with an indusium, fancy word for a cover. The indusium will retract when the spores are getting ripe. Not every genus has an indusium though. While every genus has differently structured sori, there are a few things that will help you know when to collect. 1. The spores have turned to the color that indicates ripeness for that species. 2. The indusium, if any, is at least partially retracted or fallen off. 3. Some of the sporangia may have sprung, which indicates the sori are really getting ripe. But bottom line, if you collect it and you get spores, it was ripe. And of course, these same indicators can be used to tell you when a frond has gone past its prime. I put some example photos on the website and some links to sites where you can look up what the sori look like for various ferns. But if you're out hiking, you have to get the frond home. I found the best way to do that is what you're seeing here on the screen.
And I've done all that. I've got a bunch of ferns on a table. I got all kinds of ferns and crap that I'm supposed to collect spores from. So the whole first part of my video series is going to be collecting the spores and being able to store them. I watched another video about a fellow, kind of an odd character, but he seems to know his stuff, and he uses aluminum foil to finally put the final. He uses actually he'll drop the mushroom on the mushrooms on the the aluminum foil, let them drop the spores that way, which I will probably do that way. I don't have to transfer everything. But I've got the other spores from the ferns on paper, and we'll watch the procedure as to how he drops
Get
the
a nice clean
spores.
piece of paper, you put the frond in, you fold, accumulated a pretty good load of fungus, algae, moss, and other spores that will grow in your meat.
What's he doing?
What's he doing? Plastic containers. Sometimes that can work, but more often than not, the moisture in the bag causes problems. The frond has been out in the weather for many months, and it's accumulated a pretty good load of fungus, algae, moss, and other spores that will grow in your medium if you don't get rid of them. Fortunately, the spores are inside the sporangia and they're very well protected. So what we'll do is we'll take the frond and dip it into a chlorine bleach solution. I use 1 part household bleach to 9 parts water. That's a
Let's see. Okay, so this is something I didn't do. Let's see. Frond sterilization. 1 part bleach, 9 parts, I'll just say distilled water.
(1 to 10 dilution). I leave it in there for about 15 seconds. I take it out, put it on a clean paper towel, and blot it more or less dry. The fronds are then taken and placed between 2 sheets of printer paper and allowed to sit for a few days in a draft-free place. The spores will drop off and they will stick to the paper, and then we can use them in the next step.
I didn't sterilize my first group.
Media. I use a mix of 1/2 African violet soil, 1/3 peat moss, and 1/6 turface MVP. I mix these with distilled water and put them into a 6-inch Petri dish, which will be baked at 200 degrees Fahrenheit for at least 40 minutes.
Oh, God.
The end result will be a covered Petri containing a damp but not flooded, sterile growing medium. Since water evaporates during baking, you need to add extra water to the Petri before you put it in the oven. Then bake it until the right amount of moisture is present. Alternatively, you can put the Petri dish into the oven with dry soil, cook for 30 to 40 minutes, and then add boiling water and cover. Either way, you want to put the cover on while the oven is fully heated, cook for an additional 5 minutes, turn the oven off, and let the Petris cool in the oven. By the way, don't worry if it dries out. You can always add more water and cook a little longer. Okay, your Petri dish is ready and cooled, and the frond has been dropping spores for a couple of days, so you're almost ready to sow the spores. Open up the paper with the frond and check that the spores have dropped. You'll see anything from yellow to black dust. Usually there's more than you see here, but this particular frond was collected after most of the spore had dropped. Now if you look at the dust under a microscope, you'll see that much of it is spent sporangia, and we know that the sporangia are where the fungus, moss, and other spores might be, so we need to get rid of it. We eliminate those contaminants by taking the paper, shaking it a bit, and then tilting it to let the chaff run off. If you do it right, you'll end up with something like this, where the sporangia are gone and you're left with just the spore. The final preparation step is to decide how much you want to sow, and I can't stress enough that you need way less spore than you think you do. So take some scissors and cut out a section of the spore. In fact, it's a good idea to cut several pieces with different amounts of spore on each one, and sow multiple Petri dishes so that at least one will have the right density of spore. Now that everything's ready, it's time to sow the spores. And as luck would have it, sowing the spores is one of the easiest parts of the whole process. I make a paper tube that fits just around the Petri dish, and I set it there to guide the spores down to the medium below. Then I bring the paper to the top of the tube, turn it upside down, and tap firmly on the back to release the spores. I wait a few seconds for them to float down to the Petri dish, and then I put the lid on, and the spores are sown. After sowing, the Petri dish is wrapped with saran wrap. This holds in moisture and helps prevent air currents from introducing contaminants. This gives the gametophytes, the first stage of fern growth, a head start. The saran wrap will stay on for 1 to 3 months. It only comes off once the gametophytes have covered the surface of the Petri dish with a green layer.
1 to 3 months. Wow.
After sowing, place the Petri in the light and let it grow. In a few weeks, you should see some green, and eventually the gametophytes should cover the surface of the Petri. Ideally, when the gametophytes get big enough to touch each other, they are between 1/8 and 1/4 inch in size. But even without this optimum density, you can still successfully grow ferns. This stage can last from 2 to 6 months, and you may have to open the Petri in order to add water or deal with other problems.
Wow.
Check the website for helpful hints on this stage. When the gametophytes are crowding each other in the Petri dish, it's time for patching out. Prepare a tray of sterilized soil, and using tweezers lift patches of gametophytes out of the Petri and place them gently on top of the soil in the tray. Each patch, containing from 5 to 20 gametophytes, will continue to grow until the gametophytes are fully developed and ready for the sexual reproductive stage. Spray them vigorously with boiled distilled water to put them in good contact with the soil. And then cover the tray with saran wrap to maintain high humidity, which is essential for the fertilization stage. After patching out, I usually give the tray a day's rest under subdued lighting before it goes back under regular lights. In 1 to 4 months, depending on the species and the conditions, fertilization will occur and the gametophytes will create a new plant, the sporophyte, or what we call the true fern. One tiny frond will emerge from the gametophyte and will eventually develop into the fern you've been trying to grow. Over time, and it might take months, many sporophytes will appear and grow until they develop 2 or 3 true leaves, which is an indication that a root system is also developed and you have successfully propagated the ferns. At this point, there are several choices of how to proceed, some of which are on the website. The one I like is to transplant individual ferns. As before, prepare a tray with sterilized light soil and one by one transplant the tiny ferns to the new tray. You can separate them from each other into individual ferns, or you can transplant small clumps. Which you choose depends on the species and what you want the final product to look like. After this step, they will grow like any other plant until they need to be potted up into individual pots. So congratulations, you've successfully propagated ferns from spore.
Not me, you
well,
have.
Folks, that's it. I hope this video has been helpful, and
Well, let's see.
So congratulations, you've successfully propagated ferns from spore.
Well, folks, that's it.
Okay, so this is the basic video that I followed in order to start spores from ferns. Now, what we're going to do is I'm going to block this out here. I wonder if this is the one I used here. Let me just take a look at this real quick here.
Social Security.
That's why we block this out so we don't have our ads in our videos here.
Of tiny little seed-like spores.
Okay, so
welcome to the garden.
Alright, so we're gonna watch this video. I believe this is the video that I was watching earlier, or a long time ago. So now we're talking about spores from moss.
Today I'm going to be experimenting growing moss from spores. Now in these little pods are probably thousands of tiny little seed-like spores.
Thousands?
So they just look like dust or smoke.
In one pod?
Incredibly small.
Wow. Did you see that? The dust on his finger?
This
Those
will
are
be
the
the first in a
spores.
series of experiments
That's
to
pretty
see if
cool.
we can grow moss from spores on the ground, in containers, and in terrariums. We're going to be starting a brand new terrarium just for this moss experiment. This was an old whiskey bottle. It had some really tasty Jim Beam. But we're going to start off with some charcoal and some pebbles to make a false bottom. This will create a little gap in between any standing water at the base and the soil so that it doesn't get oversaturated. I don't want to get anything that's still kind of brown like this. That means the wood isn't fully burned. So finished charcoal should be nice and light. It should be pitch black. We want to break this up into pieces about the size of our pebbles, just so they fit in the jar. Now we're going to rinse this until the water is fairly clear. This will wash away any ash, which can actually change the pH, and it'll make sure that any standing water down at the bottom isn't black and soupy. I've also rinsed the pebbles too, but that's less important. It's just going to keep that water nice and clear. By making several terrariums, I've discovered you really only need a tiny little layer down at the bottom. So we're going to shoot for about 1 or 2 centimeters, starting with our rinsed charcoal.
Now I thought, this wasn't the video I watched before. The other video I watched had the stones on the bottom. Then it had the layer of like, well, I'm using a cloth, and then the charcoal, so I don't know what's going on here.
I've made sure to save enough charcoal to mix in with our potting soil. So we've got a good amount there. Just gonna kind of spread it out a little bit, and then we can add our rocks. We could mix this up, incorporating the charcoal in with the stones, but I kind of like it as a base. It provides a nice visual anchor with that beautiful dark color, but it's also right at the bottom where any standing water will be, so that's where it's going to be most useful to us. This is going to be a native terrarium, using mosses that we've collected right here in the backyard. We could probably use native soil, but I want our soil to be nice and clean, so that if any moss sprouts, I know it was from our spores. So I'm going to be using just my normal potting soil from indoors. That way I know it's nice and sterile. And so we don't make too much of a mess, I'm going to take a little piece of paper and make a funnel. So I'll just fold it over like this with a little hole at the base.
Why not just use a regular funnel, you know? Huh.
And periodically I'm going to mix in some of our charcoal as well.
That's not even going to go through the funnel. Oh.
And we're only going to add about 2 inches of soil. Because we really don't have any plants in here, they're not going to need a lot of soil for those roots. Moss actually don't have typical roots like normal plants. Instead of roots, they have rhizoids, which are hair-like structures. Roots act like a straw, transporting water from the inside, whereas rhizoids act like a wick, drawing the moisture up. You can actually get a really good look at them in my first ever terrarium. They just cling to the side of the glass, so you get an idea of the structure of the plant. And before we add our spores, I want to pre-moisten this soil.
I like the sound of
fun
this.
Part. We get to add our moss spores. So I've collected most of these with the scissors, just to get those spore pods at the top.
Huh.
Just gonna grab a couple more here. I want to make sure I have enough for multiple different experiments. I want to do some of these in terrariums, some of them on terracotta pots. I want to try just smearing
I got a
some on
terracotta
some
pot
soil
too.
and see if we can get them to come up that way. So I don't want to just pull the spore pods out, because we'll actually pull out a lot of the plants with it. You can see we can uproot, or up-rhizoid, some of those moss plants. And even though they look thick, this is really what each individual plant looks like. They're really small, and there's really not much to them. Different varieties of moss can have different looking spore pods. These ones kind of look like little figs. I do want to be careful not to introduce too much moss. That can get rehydrated and might give us a false positive. So I want to try and stick to just the spore heads. And we can actually break these apart and kind of sprinkle them like dust. They just look like dust. They are absolutely minuscule. So there's probably a million on my finger right now. And while we're adding these, I want to try and break them up a little bit, just so that they spread out a little more evenly.
Huh.
So this is all an experiment. I've never actually gotten moss to grow from spores before. Now, other than moss to grow, or if we get any oven. Moss needs a surprising amount of light when grown indoors, so pick a really bright window. And this is just the first of several experiments on growing moss from spores. So I hope you stay tuned. Thank you all so much for watching, and we'll see you next.
Growing moss from spores. 2 days ago.
So I hope you stay tuned.
So that was like
Thank you all.
August, September, 3 months ago. You guys got a lot of videos. Oh, I think I tried to look for this and I couldn't find. Okay, there it is. I don't think there was a result. I don't know. Maybe there was another. It's growing ferns. Moss life cycle under a microscope. Let's see what that is.
Social Security is threatened long.
Oh, we don't want his face on my video. Good God. $150 magnification. $1,500? Wow. Holy shit. I don't care about the damn music. That's a live picture. There was all stuff moving around in there. So I guess that's pretty much it, I think. Collecting and using moss. Growing moss in containers, I think.
Plant out some moss. It's not a typical plant, and they don't have roots like a typical plant.
Roots.
If we can get a close-up of this, they just grow on the surface of soil, and they make a nice thick carpet where they got quite a decent amount of sun. This one was on a rock day, and I can see actually that these little tree-like ones that were super dry are now actually starting to plump up. So they're quite resilient, and that shriveling up was a water-saving method, but they plump right back up.
So my problem is I've got 2 weeks worth of stored mushrooms and ferns and moss in bags, and I've got to try to bring all that stuff back to life. That's going to be the goal. I've got some things that are in the freezer, which I'll be doing some of that. I'm going to be splitting up spores and moss from growing from regular temperatures to frozen temperatures. And as far as seed germination, I've read where if you put seeds in the freezer for a certain time and then try to plant them, they will germinate quicker. So we'll be doing an experiment on that. Why I'm doing all this, I don't know. I do not know. What's a succulent?
In 2000.
Oh my god.
Colder climate or in winters, it gives much better result. Today we will propagate from callus and are ready to be propagated with this. See. Take a shallow tray with drainage hole in the bottom and fill it with cactus mix, or you can make your mix by yourself by adding sand, cocoa peat and perlite in equal proportions. But I have got bad. Guys, have a happy experience and grow more and more succulent from this easy simple way.
Yep. Making tiny native terrariums. I thought there was another video that I watched. How to grow moss. How to propagate moss. Moss releasing spores. Ooh, here we go. Huh. I didn't see any moss. Oh, I see it. It's right there at the bottom. It was right at the beginning of the video. In the middle. The middle one. Right at the very beginning. It dropped some spores. Pretty cool. Well, that's enough for tonight, I guess. My next video, hopefully, I'll be able to work with these different things, and I might go through some of those sterilization processes that I'm using, and then I'm going to try a different growing medium rather than using agar. I've got gelatin. We're going to try that. Yep, I am done. Thanks for watching.